Hi folks, welcome back to the stats section of the dyspepsia module. I'm Tom Reeder and I'm going to be talking to you today about differences. So this is the second of 3 lectures I'm giving you on stats, and the idea is to try and prepare you for handling data sets and testing some ideas about those data as we go along. So today we're going to look at differences and how you might test for differences. So I'm an ecologist, so I am fond of illustrating concepts with ecological pictures. So what you can see on screen here is 2 snakes, which just about superficially, if you squint at them, look rather similar. Brightly coloured stripy snakes. One of them is deadly, the other is completely harmless and is thought to mimic the other one, pretend to look like it in some way with those red bands. And telling the difference is rather important because it could save your life. So differences in all kinds of scientific contexts are important, and we're going to look obviously today at sort of rather more relevant examples than this to a pharmacy degree. So the outline for today is to start off by actually revisiting what we were doing last week. I took you through a kind of thought exercise looking at the plague and a possible treatment for the plague, which introduced ideas about hypotheses and how we test hypotheses, and ultimately led to us generating, without really realizing it, something called a p-value. So we're going to look back at those ideas and formalize what I told you last week. And then we're going to look at the situations, the common situations in which we actually use p-values to test ideas. And there are really 2 situations: when we're looking at differences and when we're looking for trends. And then for the rest of the lecture, we're going to focus in on differences. Trends we'll deal with in more detail next week. But we're going to look at how to test for differences using a couple of different tests, the t-test and the chi-square test, both of which might be familiar to you if you did any stats at A-level, but don't worry if they're not. We're going to take you through how those 2 tests work. So part 1 of today's 3-part lecture is to look back at what we did last week, think about what is a hypothesis, what is a p-value, and how we can use a p-value to tackle a test of a trend or a difference, or at least how we can use it to reach an objective decision about a hypothesis. And then we're just going to have a start to look at differences and trends as important patterns in science, and how we might actually look to detect objectively whether a difference is present or not. So going back to last week then, I gave you this example of a treatment, a poultice that you could put onto a patient's body in an attempt to cure them from plague. A genuine idea from the 17th century, but rather implausible by modern medical standards, to smear somebody in tree resin and human feces. But hey ho, they had to try something. And I imagined a situation in which we collected a bunch of data in the form of patient outcomes who'd been treated with this poultice, and we discovered that of all the patients who had plague who were treated, they all died. And I talked about the fact that the patients that we chose from the wider population of people with plague represented a sample, and because we took that sample at random, hopefully, that sample was truly representative of the underlying population. So the words population and sample are really important bits of statistical jargon that you need to remember. The population is the whole group of individuals that you're trying to make generalizations about. The sample is the small subgroup that you have access to and from which you can collect data. So key concept here with a population is that it represents all of the individuals in which you're interested. That isn't necessarily all of the individuals of that type in the world, I suppose, in the sense that, for example, your population might be all of the cells in a petri dish, or all of the petri dishes in a laboratory. It doesn't necessarily have to be all of the cells in the world. It depends on your research question. So the key thing is, what are you trying to research? What are you trying to ask a question about? If you're trying to ask a question about all of the cells in this particular human person, then that's your population. If you're trying to ask a question about all of the liver cells in the population of people living in Nottingham, then that's your population. The sample is a representative, normally random, selection of individuals from that population that you've defined previously. So if your population is all of the people who have plague in London, your representative sample hopefully would be selected at random in some way from the people who have plague. I say in some way, obviously in many situations it's actually quite difficult to choose a truly random sample, but you must do your best as a scientist to come up with a sample that represents, it's not a biased sample, it's a representative sample of the population, in order that you can reach an objective conclusion about the population from the sample. So this is the principle. The reason we need a random representative sample from the population is we want the sample to represent the population, so that when we make a conclusion about the sample, we can extend that conclusion, generalize that conclusion to the whole population. So in our plague trial, what we wanted to do was use a small number of people with plague to tell us something about the wider population. Would they get better if they were treated with this poultice? So we also generated, as part of this thought experiment, effectively a hypothesis, and more than that actually, we generated a pair of hypotheses. This wouldn't have been clear to you last week, but let's look back at what we did. We treated patients with this poultice made of tree resin, lily roots and human excrement, and we postulated that the poultice would alter the chance of death. Hopefully it would improve the chances of survival, reduce the chances of death. But we did also recognise that the opposite was possible. Smearing somebody with human faeces might actually reduce their chances of survival. But nevertheless, at the beginning of the experiment, what we did was we said, well, we think the poultice might change the chance of death. That effectively was what we call the hypothesis, or sometimes specifically the alternative hypothesis, H1. So it's the statement, the postulation if you like, about your study system, which says what you think might be happening. And it always comes with a natural kind of pair called the null hypothesis, which is the opposite, which is a statement that says nothing is happening. So in our case, we knew that before we started the experiment that in the natural population who've got plague, about 50% of the people will survive and 50% will die. So the null hypothesis was that applying the poultice to patients wouldn't change that probability of death. So the poultice will have no effect on the probability of surviving or dying. And that's the null hypothesis because it's framed in a negative way to say we don't think that anything will happen. We don't think there will be any difference between the two groups of patients, those treated and those not. And we abbreviate it to H0 or H0. So H0 and H1 are a pair of hypotheses. One says something's happening, one says something's not happening. And it's these two hypotheses that we try to challenge with data, and we try to reach a conclusion about which of them is more plausible. So these are key concepts. The idea that a hypothesis is not just any old statement about what's going on, it's a specific statement that something is happening in your study system. That's H1, the alternative hypothesis, versus a statement that says nothing is happening, which is the null hypothesis. So typically in pharmacy, the alternative hypothesis might be of the form that drug X reduces symptom Y. So you're saying what you think is happening or might be happening, as opposed to a null hypothesis which says drug X does not reduce symptom Y. Okay. What else did we do? What do we do with these hypotheses that we had that we didn't really articulate in that way, but that were clearly present in the thought experiment we did? Well, what we tried to do was collect data in the form of patient outcomes and decide at what point we got enough data that we were convinced the poultice was definitely changing the chances of survival, or was definitely not having any effect on the chances of survival. And I asked you, well, if you were in the lecture theatre, I would have asked you to put your hand up. I asked you to make a mental note of the point at which you felt you were confident you understood the outcome of the trial, you had enough data to reach a conclusion. At that point, what you were effectively doing was saying you had enough evidence to either accept or reject the null hypothesis. So the null hypothesis said there is no effect of the poultice. And eventually, we'd seen enough dead bodies that I think all of you will have concluded there's enough evidence that that null hypothesis is wrong. The null hypothesis says the poultice doesn't have any effect. The data suggested, yes, it does. It kills people, because instead of seeing 50% of people dying, we were seeing everybody dying. So we rejected the null hypothesis and retained the alternative hypothesis. The alternative hypothesis said the poultice has an effect, and it looked a lot like it did. And we reached that conclusion once we got so much data. So, well, why did we stop at that particular point? And I illustrated what was going on by calculating these p-values, these probabilities, the probability that what we were seeing was happening by chance in a world in which 50% of people die of plague. So that was our null hypothesis, that 50% of people will die from the plague regardless of whether they're treated with the poultice. And what effectively we did in our minds as we were thinking about the data was to work out how unlikely it was that the data we were seeing was consistent with that worldview, that the poultice had no effect. And when we got to the point where we thought it's really unlikely that the poultice has no effect, it's really unlikely that the null hypothesis is correct, it's at that point that we stopped the trial. And we stopped the trial because we had reached a conclusion, right, we don't need to kill any more patients, we need to stop using this poultice and try and find a better cure. So what you were doing mentally was kind of estimating this thing we call the p-value, the probability of the data under the null hypothesis. And scientists have generally agreed that there will always be some probability that the p will never equal exactly 0, because even extremely weird events in the natural world could happen by chance. But once that p-value gets really low, it gets to a point where we can effectively ignore the null hypothesis, reject it, and instead conclude that the alternative hypothesis is correct. And that threshold, the point at which we make that decision, we can quantify it as a particular p-value. And the standard p-value we use is 0.05 or 5%. And that threshold p-value, 0.05, is called alpha. So alpha is a particular p-value which represents the threshold for decision making about a statistical test, about a data set that we've got. When the probability drops below alpha, typically 0.05, not always, sometimes alpha is something different, but when alpha is 0.05, if the p-value drops below 5%, 0.05, then we decide there's enough evidence to reject the null hypothesis. If the p-value doesn't drop below alpha, we conclude that there isn't enough evidence to reject the null hypothesis, and there isn't any evidence supporting the alternative hypothesis. That's how we do stats tests. So we were doing all of this in our minds without any need for this jargon and Greek letters like alpha, but actually it helps if we formalize it in those ways in order to understand more complex situations. So absolutely critical concept. The most important thing I'm teaching you in all of these lectures really is the p-value. It is the probability of the observed data if the null hypothesis is true. So what are the chances of that happening if the null hypothesis is really true? And alpha is the significance threshold below which we need to see the p-value fall if we are going to reject the null hypothesis. Because what we're looking for is data that are so unlikely in a world in which the null hypothesis is true that the null hypothesis is probably false. So if you like, alpha is how unlikely is so unlikely that it cannot be that it can be ignored. And as I say, alpha is typically 0.05, but sometimes it's set at 0.01 or a 1% chance, and sometimes even a 1 in 1,000 chance, 0.001. Where you set alpha before you start collecting data is important, obviously, because it can affect the outcome of your test, and it tends to reflect how conservative the scientists are being about their particular study system. So if you're looking at treating patients with something that may be a drug that is potentially dangerous or has nasty side effects, you're going to want to be very, very confident that any positive effect you see on patient outcomes is real. And so in that situation, you might adopt a very conservative alpha threshold, a very, very small number, like say 1 in 1,000, because you don't want to risk getting it wrong. Whereas perhaps in a less critical situation, you might adopt a less conservative alpha value. And like I say, as normal, in normal scientific studies, it's typically set at 5%, 0.05. Okay, so I hope what we've demonstrated with that kind of thought example and then putting the jargon on the example is that stats tests are really useful. and their main job is to calculate this thing called P. And P tells us objectively, instead of having to stick a finger in the air and look at the data and kind of go, yeah, it looks like there might be a pattern, it tells us objectively how likely the data are if the null hypothesis is true, and therefore it allows us to make an objective judgment about whether the hypothesis, the alternative and the null hypotheses are true. So hopefully we transition from being, if you know your Simpsons characters, kind of dodgy, make it up as you go along pharmacists, to gold standard, good scientific brain, objective scientists. Right, so we nearly finished this little part of the lecture. What we're going to do now is look at how we can use that whole framework of hypothesis testing and generating p-values to test real sorts of patterns that we might be interested in your case in pharmacy. So the sorts of situations that we're typically looking for fall into two categories. Either we're looking for a trend or we're looking for a difference. On the left hand side here, typical situation in which we're looking at the relationship between the dose of a drug and some kind of response to that drug. In fact, we've got two different drugs and then the drugs in combination, so different scenarios. But essentially what we're looking for is how increasing the dose changes the response in the patient. When we have a relationship like this between two continuous variables, the concentration of a drug in this case and some measure of a response, doesn't matter what it is, if they're two continuous quantitative measures, variables, then what we're looking at is a trend. The alternative scenario that's very common in science is to look for a difference. So where you, instead of having two continuous variables, you've got one categorical discrete variable like this one here. So this is whether or not people have consumed alcohol, yes versus no, and then how many hours they slept. So here we have a quantitative continuous variable and here we have a categorical variable. So when we're interested in looking at a difference, in this case between people who have had alcohol and people who have not, in a continuous response variable like the number of hours of sleep they've had, then what we're looking at is a difference. So difference versus trend, two different kinds of patterns that we might be looking for in data. And for the rest of this lecture, for the remaining two parts, I want to look specifically at differences before next week we move on to looking at trends. So in the case of a difference study, essentially what we're talking about is a study in which there's a hypothesis that makes some kind of statement about a difference between experimental samples or groups or treatments, often a control versus a drug, for example, and we want to know whether there is evidence that there is a real difference. So kind of classic example from the real world, or the kind of outside the laboratory, if you like, is Head and Shoulders man. This guy is apparently, in fact, experiencing two different treatments here. We've got the guy at the top who appears to have dandruff in his hair, and the guy at the bottom who appears to have lovely glossy hair with no dandruff. And this is because the guy at the top comes from a sample that's been is a control sample, and the guy at the bottom comes from a sample that's been treated with Head and Shoulders shampoo, which allegedly reduces the appearance of dandruff. Whether or not we believe what we can see in this photograph, I'll leave to you. And in fact, if you zoom in on that image, you'll find that those are photoshopped pixels of dandruff, but let's not worry about that. But they illustrate a point that we're often trying to take individuals, expose them to two different treatments, control and Head and Shoulders, for example, and then collect some data, in this case about dandruff, and find out if there's a difference. And that's what we're going to spend our time looking at in part 2 and 3.